Moving on, we will explore example and applications of tissue culture. Tissue culture is a method which fragments of tissue from an animal or plant are transferred to an artificial environment in which they can continue to survive and function, such as agar or liquid medium, rich with nutrients and hormones. There are many techniques in tissue culture, but we will only be looking at two techniques for plant and animal tissue culture. For example, in plant tissue culture, we can introduce a plant seed into a growth media to generate seedlings or plants. It is the best method for raising sterile seedlings. If you look in the picture, they introduce the seeds into the culture, and after a few weeks, the seed will grow into seedlings which then can be acclimatized into the proper or natural environment and then transferred to the field. Another method of plant tissue culture is meristem culture, in which a small piece from the shoot apical meristem is obtained, sterilized, and transferred to a culture. The resulting clump of cells called callus is then broken up into different set of cells, into individual cells, and it will grow into different set of plants. Meanwhile, for animal tissue culture, techniques that can be employed are organ culture and cell culture. For organ culture, a piece of the extracted tissue is introduced to a medium, which will grow into an organ. Again, the medium is rich with nutrients and hormones that will help the extracted tissue to grow. In cell culture, instead of tissue pieces, single isolated cells, which are normally suspended in a liquid medium first, only then it will be transferred to a medium and incubated for growth.